in which Jung will defend the academic thesis Olfactory System Vulnerability in Alzheimer's Disease, a multidisciplinary study decoding Alzheimer's through the nose-brain axis. May I invite you to present a summary of your study and the conclusions of your thesis, and you have approximately 15 minutes.
Thank you, Prorector. Dear Prorector, dear members of the Corona, dear families, friends, colleagues, and those watching us online, in the next 15 minutes, I will present the main findings of my PhD thesis entitled Olfactory System Vulnerability in Alzheimer's Disease, a multidisciplinary study, with the subtitle Decoding Alzheimer's through the Nose-Brain Axis. This research explores how the sense of smell can provide a unique window into the earliest stages of Alzheimer's disease, from molecular alterations to clinical translation. I would like to start by introducing you to Alzheimer's disease. Alzheimer's disease is the most common cause of dementia and is characterized by both structural and molecular degeneration in the brain. On the left, you can see how the cortex and hippocampus shrink as neurons die, while abnormal proteins, amyloid beta and p-tau, accumulate as plaques and tangles, which are the classical hallmarks of the Alzheimer's pathology. On the right, PET scan images illustrate disease progression from healthy aging to mild cognitive impairment, in short MCI, and Alzheimer's dementia. As the disease advances, regions responsible for memory and thinking become progressively less active, leading to the cognitive symptoms we observe clinically. These brain changes manifest as gradual loss of cognitive and behavioral abilities. Early on, patients may experience mild forgetfulness or misplacing objects. As pathology spreads, difficulties in decision-making, language, and orientation emerge, and ultimately, patients lose independence in daily life. Alzheimer's disease therefore affects not only memory, but also personality, mood, and behavior, fundamentally changing how a person interacts with the world. Interestingly, one of the earliest and most consistent symptoms of Alzheimer's disease is the loss of smell. Many individuals show measurable olfactory decline years before any noticeable memory impairment. These early changes reflect underlying molecular events, the buildup of amyloid and tau within the olfactory bulb and olfactory cortex. As shown here, olfactory dysfunction often precedes both brain structure changes and clinical symptoms, highlighting its potential as an early indicator of disease progression. To understand why the sense of smell is so closely tied to Alzheimer's disease, we must look at the anatomy of the olfactory pathway. The olfactory system is unique in that odor signals directly reach the cortex, bypassing the thalamus. Information travels from the olfactory epithelium in the nasal cavity to the olfactory bulb, then to the olfactory cortex, amygdala, and hippocampus, regions responsible for memory. This process can be thought of in three stages: detecting, encoding, and processing. During the detecting stage, sensory neurons in the nasal cavity capture odor molecules and translate them into electrical signals. In the encoding stage, the olfactory bulb acts as the first relay, refining and organizing this information into distinct odor patterns. Finally, during processing, these signals reach higher cortical and limbic regions where they become integrated with memory, emotion, and perception. This smooth flow from detection to processing helps us connect smells with memories. But it also means that even small disruptions at the first stage can affect broader memory functions. Because of this direct connection, olfactory regions are not only central to sensory experience, but also particularly vulnerable to early Alzheimer's pathology. This anatomical flow from the olfactory epithelium to the bulb and to the cortex also forms the structure of my presentation today. Now that we've seen how the olfactory pathway connects the nose to the brain, this brings us to the central question. How does Alzheimer's pathology emerge and propagate along the pathway? To address this, I structure my research framework along the nose-brain axis, covering the olfactory epithelium and the bulb and the cortex. At the level of olfactory epithelium, we analyzed human nasal discharge fluid to investigate how peripheral A-beta-42 levels relate to brain amyloid burden and cognitive status. In the olfactory bulb, we studied human postmortem tissue for amyloid, tau, and glial pathology, and mouse tissue for amyloid accumulation to explore region-specific vulnerability across species. Finally, in the olfactory cortex, we examined human postmortem tissue for amyloid, tau, and glial changes across cognitively normal, MCI, and Alzheimer's cases. This integrated design allows us to compare molecular and cellular alterations across multiple disease stages within a single sensory system that extends from the periphery to the brain. Here, we examined the olfactory cortex using human postmortem tissue across 3 clinical stages: cognitive normal, MCI, and AD. As you can see in the representative images, amyloid beta in magenta and phosphorylated tau in green show a clear progressive increase, particularly evident in Alzheimer's disease cases. These findings demonstrate that the olfactory cortex undergoes stage-dependent accumulation of amyloid and tau, highlighting its critical involvement in Alzheimer's pathology. While amyloid and tau aggregation represent the major hallmarks of the disease, they do not act alone. To understand what drives or amplifies these changes, we next focus on the brain's immune environment, specifically glial cells such as microglia and astrocytes. Beyond protein aggregation, Alzheimer's disease also involves complex inflammatory processes. In the healthy brain, microglia act as immune sentinels that clear debris and support neurons, while astrocytes provide structure and metabolic support and help maintain the blood-brain barrier. In Alzheimer's disease, these normally protective cells become reactive. Microglia release inflammatory mediators, and astrocytes lose their homeostatic roles and begin producing factors such as ApoE and complement proteins that can worsen pathology. Sustained activation of these glial cells amplifies inflammation and neuronal stress, accelerating neurodegeneration. Understanding these glial mechanisms was therefore central to our postmortem investigation of the olfactory system. Having introduced the role of glial cells, we next examined how they change in the olfactory cortex across the Alzheimer's continuum. On this slide, you can see representative images for microglia on the left and astrocytes on the right. It's shown for cognitive normal, MCI, and AD cases. In the MCI and AD tissues, microglia become more numerous and appear visibly activated, clustering around regions of amyloid and tau pathology. Similarly, astrocytes show stronger fluorescence and larger, more complex shapes, reflecting their activation during disease progression. Together, these patterns illustrate a clear trend of progressive inflammation within the olfactory cortex, paralleling the accumulation of amyloid and tau shown earlier. After examining the cortical end of the olfactory pathway, we next turn to the olfactory bulb, the first relay station for other information entering the brain. In this region, we analyzed human postmortem tissue for amyloid beta, p-tau, and glial markers, and examined OB tissue from the 5xFAD mouse model, which primarily represents amyloid pathology. Because 5xFAD mice develop only A beta deposition and not tau pathology, these data provide insight into amyloid-driven mechanisms, but also highlight the limitations of animal models in capturing the full spectrum of Alzheimer's disease. This makes human postmortem validation especially important. In the human samples, we observed clear stage-dependent increases in A beta and p-tau from CN to MCI and AD cases. In the 5xFAD mice, amyloid accumulation followed a similar pattern, confirming that OB is also affected in experimental models of the disease. Together, these findings identify the OB as a region of pronounced and conserved vulnerability, supported by both human and animal evidence. Having established the presence of A beta and p-tau pathology in the OB, we next examined glial activation within the same region using human postmortem samples. On this slide, you can see microglia on the left and astrocytes on the right, shown across CN, MCI, and AD cases. Both cell types show clear evidence of activation. Microglia become more abundant and clustered, while astrocytes show stronger signals and more complex shapes, reflecting reactive changes. Together, these results demonstrate that the OB is a regionally vulnerable inflammatory hub where glial responses amplify local pathology. Finally, we examined the olfactory epithelium, the peripheral starting point of the nose-brain axis. Using human nasal discharge fluid, we measured A beta 42 levels across CN, MCI, and AD participants. Interestingly, the pattern was non-linear. Rather than simply increasing or decreasing, individuals with moderate nasal A beta 42 levels showed the strongest association with both brain amyloid PET signals and cognitive decline. This finding provides crucial evidence for the nose-brain axis, showing that biochemical changes detected at the periphery can mirror ongoing amyloid dynamics in the brain. It also highlights the olfactory epithelium as an accessible and non-invasive site for monitoring central Alzheimer's pathology through peripheral sampling. In summary, these findings have clear translational relevance. The olfactory system provides an early and accessible window to the brain, allowing us to detect disease-related changes through both central and peripheral measures. Because Alzheimer's disease is largely irreversible, early detection becomes crucial. The ability to monitor changes through simple and routine nasal sampling offers easy access not only for clinicians and researchers, but also for patients and the general public. By combining animal, clinical, and postmortem evidence, our work bridges basic mechanisms and clinical application, showing how nasal and brain pathology are closely connected. Ultimately, this approach points towards non-invasive and widely applicable strategies to study and monitor Alzheimer's disease in its earliest stages. To conclude, our results reveal how Alzheimer's pathology develops along the nose-brain axis. In the olfactory epithelium, we found that nasal A beta 42 dynamics mirror brain amyloid activity and cognitive status. In the olfactory bulb, human postmortem analysis showed amyloid, p-tau, and glia activation, while mouse studies confirmed parallel amyloid accumulation, together pointing to region-specific vulnerability. And in the olfactory cortex, we observed progressive increases in amyloid, p-tau, and glial activation across disease stages. Altogether, these findings outline a continuous pathological pathway along the nose-brain axis, linking peripheral changes with the central mechanisms in Alzheimer's disease. Thank you.
Thank you very much for this clear and nice presentation. The opposition, they're all there. The opposition will be opened by Professor van den Hoven. He was the chair of the assessments committee, and he's a professor in neuroepigenetics at the Department of Psychiatry and Neuropsychology at Maastricht University. Professor van den Hoven, the floor is to you.
Thank you, dear prorector. Dear candidate, let me first of all compliment you for this very nice thesis, and also I think for this very clear presentation, and also my compliments towards your promotion committee. I do have some questions though, of course. My first question would actually be on a comment that you also state in the introduction, that there's of course other non-invasive markers, including blood-based biomarkers. And then you state that the variability in those blood-based biomarkers is one of their biggest limitations. So my question is, how do we now interpret your findings in view of that remark? So do you have any idea whether the variability that you observed in your nasal biomarkers, how they relate to blood-based biomarkers?
Highly esteemed opponent, thank you for your kind words and question. So if I may restate your question, you'd like to know how these olfactory biomarkers relate to other blood-based biomarkers. And although we did not compare the results from the blood-based biomarkers with our nasal results, our studies suggest that since our results demonstrated clear association with brain amyloid PET results, and we now know that the brain amyloid PET results and blood-based biomarkers correlate very efficiently, we can confer that our results also may be a peripheral reflection of blood-based and brain amyloid PET status.
Yeah, and as you said, you didn't measure it in the same sample, so that could have sort of shed even more light on this. But you're confident that they would at least be as good then. Okay, then let's briefly move on. Actually, my sort of second series of questions is related to the data presented on page 129, where you actually show behavioral data, and in this case, particularly in the cognitive domain, when comparing your 5-fold familial AD model with wild-type mice. And I was wondering there, also in the rest of the results section, I don't see the actual memory data, if you get my point, because everything you show here is comparing your transgenic animals with the wild types. So, for example, I cannot conclude whether there is actually intact memory in certain cases, because you only show the ratio, and also in the other data displayed, I could not easily trace this back. So, could you maybe shed some light on this? And maybe another question then is, why did you display the data like this?
So you're asking about the chapter 4 of my thesis of the.
Just as an example, if we look at the Y-maze data, where you look at the alternations made, yeah, you then compared your wild-type animals with the transgenics. You also want to see whether there is actually intact memory, at least in the wild-type animals, to show that the test is working. So in that respect, you normally compare the data to the 50% chance level. Did you also do something like that in addition to the current analysis, or the way it's currently shown?
So I think in one of the in figure supplementary 1, we showed the verification of early stage AD phenotype in 5XFAD transgenic mice, and we showed that wild type and FAD mice did not have any significant difference in number of arm entered in Y-maze series. So it shows that cognition is intact in 5XFAD mouse at this early stage, but it showed that in bird food test, it showed significant increase in time latency in bird food test, showing that when the cognition is intact, but the olfactory dysfunction precedes this cognitive impairment.
Okay, yeah. Yeah, let's leave it at that for the moment then regarding these data. Although I think it would have been helpful if you would display the actual cognitive data, because this is always a ratio and it doesn't necessarily say whether memory is intact or not in both the genotypes, basically. Although it gives you a nice overview of what happens over time. But maybe related to that, I was wondering on the breeding design used. If I look at the description in this chapter about the breeding design, then actually you have your 5-fold AD animals, and the wild types have been ordered separately, if I understood this correctly. So is there a specific reason why you did it like this? Because you can actually also get your wild type animals from the same breeding, which then corrects for all kinds of also genetic variation independent of the familial mutations, but also for differences in environmental conditions. Some might have been transported from the company where you ordered them from, the others have been used in the facilities more. So did I understand it correctly that the wild type animals and the transgenics are actually independent from each other in terms of breeding? And I got this, I think, from page 92, where you state that that they are separate.
I wish I could answer your questions now, but at the moment I don't have exact answers. So if time allows, I will get back to you later, yes, with the answers about the breeding of these animal models.
No problem. The reason why I'm also addressing this is that there has been a recent paper in Neuron showing that it even differs whether the mother is wild type and the father is then a transgenic or the other way around. That even something as simple as this may have a huge impact on pathology in these animals. Anyway, let me now move on to a different topic.
Professor van der Hoven, I think keep it short. We have to move on.
Yeah, that's absolutely fine. Maybe then briefly on you, showing that this is an early alteration in Alzheimer's, so changes to the olfactory system. So how does that, to your opinion, link to causality? Because if you, of course, look at the brain area involved in the later stages of disease, you're particularly looking at the consequences of pathology. How do you see that in view of your changes at the level of the olfactory system? So
if I may restate your question, you'd like to know whether the pathological process spread from the olfactory cortex to the bulb or vice versa, about the directionality?
Yeah, and what knowledge here can help, independent of the biomarker issue. What can you do with this knowledge in terms of causality and maybe developing strategies to intervene?
Okay, so it was our study was cross-sectional, so we cannot directly infer temporal directionality, but based on anatomical organization, the OB, since the OB is the first relay from the periphery and it directly expands to it's directly exposed to environmental factors, and it also has very high metabolic turnover and synaptic turnover, so it has very high metabolic stressors. So it makes it very plausible for initial site of pathology. But on the other hand, we also see very strong feedback from the cortex to the bulb. So I think it can reinforce this bidirectional manner. So in translational viewpoint, we can use olfactory system at the periphery so that it can reflect the central amyloid pathology as a means of very non-invasive and accessible window.
Okay, thank you very much. I'm very happy to answer. So give the word back to the PROAC.
Yeah, thank you. The opposition will be continued by Professor Goran Cimic. He was a member of the assessment committee and he is a professor in neuroscience and neuroanatomy at the University of Zagreb. Professor Cimic.
Thank you, dear Prorector, and thank you, candidate, for your excellent presentation. My first question relates to your emphasis on non-invasive bodily fluids like nasal discharge, tears, saliva, urine, that could serve as biomarker sources for Alzheimer's disease, because they are more easily collected than cerebrospinal fluid or blood. Given this fact, what would be the key methodological and biological obstacles that must be overcome for these to match the diagnostic accuracy of cerebrospinal fluid or imaging? Yes.
Okay.
This relates to panels that you mentioned. This relates to the sensitivity, specificity, falsely positive results, falsely negative results, concentrations, blood-brain barrier issues, sample variability, etc., etc.
Highly esteemed opponent, thank you for your kind words and your question. So if I understand your point, you're asking why our olfactory system is the one I chose to study and how it relates to other conventional biofluid studies such as CSF or blood-based biomarkers. The main reason is that olfactory system provides both functional relevance and anatomical continuity with the brain. But unlike CSF or blood, it directly connects the periphery and the central nervous system via the olfactory bulb. And pathology in these regions directly affects sensory function. So, and moreover, as I mentioned earlier, nasal fluid sampling is non-invasive and repeatable, which is a very key point when we choose to translate them into real world so that clinicians or researchers or even the patients or the public at risk can use this routine sampling as a means of early detection or early diagnosis. So while CSF and plasma remain essential for quantification, but the olfactory system allows us to study where and how pathology originates.
Okay. My second question would be, what mechanisms do you think might link region-specific accumulation of amyloid to selective olfactory neurons dysfunction? Because amyloid, when soluble, is highly protective. And what would be the consequence of anti-amyloid therapy?
So thank you for your question. So, and if I may restate, you're asking how this microglia in the olfactory bulb can influence the olfactory, can influence the pathogenesis in this olfactory system. And
if I may interrupt you, opinion leaders in the field, they think that amyloid pathology and tau pathological changes evolve separately over long periods of time for decades. And then due to physiological inability of physiological processes of clearance of misfolded proteins and strains, after this failure, a sort of tau influences amyloid and amyloid influences tau pathology. And then this started to change more rapidly. In this context, where would you put the olfactory system in respect to other systems in terms of like Bragg criteria? So how early you think that olfactory changes occur in respect to other systems?
so from our animal studies model in 5XFAD mouse, which only shows amyloid beta pathology, we saw that amyloid accumulation in early stages, and we also saw olfactory dysfunction in the absence of tau pathology. So we think that in amyloid alone pathogenesis, it can drive the olfactory dysfunction. And also the view in the popular view of this field is that when amyloid is the driving force, then and the tau is the actual killer of these disease stages. So yes, tau can in somehow maybe accelerate the pathogenesis, or it can worsen the neuronal degeneration. We think amyloid is capable of driving this olfactory dysfunction in olfactory system.
My last question. All of your findings, they point to...
Keep it very short, because we are running out of time a bit.
Okay, this is my last question. So there has been increased understanding of microbe or infectious origin or of several different neurodegenerative diseases. So particularly for Alzheimer's disease, herpes simplex virus 1 is perhaps the most commonly mentioned. How do you think this virus can trigger changes? And do you happen to use some markers for either microglial inflammasome, NLRP3, or neuronal and NLRP1 to show sort of this silent neuroinflammatory changes induced by microglial cells as well as astrocytes? Have you got any hint on those?
Thank you for your questions. So you're asking whether I've done any experiments to study if there's any virus infections that could induce
either in fluids or in tissue.
Unfortunately, we did not run any virus-related experiments, but since the olfactory epithelium is in direct contact with the external environment, and maybe airborne toxins, pathogens, and viruses can reach the bulb via the olfactory nerve. And other experimental studies have shown that particulate matter and viruses can induce local inflammation and microglia activation in this region. So while we did not directly prove any causation in viral infection and the olfactory dysfunction, it is possible, it is plausible that chronic exposure to these pathogens or viruses may prime local immune cells and thus making the olfactory bulb more reactive to Alzheimer's pathology. And I think this environmental interface could partially explain why olfactory regions are affected so early in this disease.
Would you confirm then that this is a multifactorial disease in that sense?
Yes, that is very possible.
Thank you.
Okay, then we move on. The opposition will be continued by Professor Martinez. She was a member of the assessment committee and she's the secretary of this degree committee. And she's a professor in neuroinflammation of neuropsychiatric disorders at the Department of Psychiatry and Neuropsychology at Maastricht University. Professor Martinez.
Yes, thank you, dear Prorector. I would like first of all to congratulate the candidate for the very interesting research thesis, and also for the of course the promotion team that has supported the candidate. I have several questions for you today, especially I am going to focus in chapter 4 and chapter 5. If I will start in chapter 4, I would like to know from you, because you have very well highlighted in your presentation that you have correlated your findings between the human olfactory system and also the olfactory system in the mice, in the Alzheimer model that you use. So when you look to all the data you have, there are some discrepancies. So how do you interpret these discrepancies, especially regarding the amyloid accumulation, which seems to be some differences in between the human findings and the AD model? Could you elaborate on this?
Thank you for your highly esteemed opponent. Thank you for your kind words and the question. So if I may restate your question, so you would like to know if I can explain the discrepancy from the animal mouse model results and the human mouse model results. So yes, and that is why we focus on the human postmortem samples more, because as you know, there's interspecies differences and the mouse model has limitation of only showing A beta pathogenesis. So, but nevertheless, human tissue provides clinical and pathological realism, but we only saw static information since we used the cross-sectional studies. And the 5XFAD mouse, it allowed us to study amyloid-driven vulnerability in a very controlled environment. And as I mentioned—
Yes, sorry to interrupt. When you look to the age of the animals and the age of the samples from the patients, individuals that you analyze for the brain, so do you think there could be a discrepancy in terms of how long they have been ill when you do the analysis compared to the animals where you have perhaps analyzed other ages? Could you perhaps also elaborate about that?
Oh, yes. So we use very young age models in mouse models, but we used old humans samples in postmortem samples, so we cannot compare the results on the side by side. But from our animal models, what we draw conclusions from animal model studies is that in early stages, before the cognitive symptoms, we saw that olfactory dysfunction may come earlier than the cognitive symptoms, and
Yeah, that's clear. So I would like to bring to the next question. So you think the beta amyloid accumulation in the olfactory epithelium is the cause of the damage to the neuron axons? Or how do you think the mechanism is why the beta amyloid in this case, in this chapter, might be driving the pathology? Could you elaborate about that? So do you think it's coming from the outside, so the pathology there is amyloid that will block connectivity between neurons, or do you think this is something, so this is just the garbage, the amyloid is going out because it's already accumulating too much inside and goes out, and it's a survival mechanism. So could you elaborate a little bit about how the beta amyloid accumulation causes the damage, if it will be causing the damage on the olfactory system? How does it work? Why?
Okay, so if I understand correctly, so you'd like to ask how amyloid accumulation is causing neuronal damage, and it's causing
In
the
your system, yeah, in the
This is-
olfactory system, yes.
Okay, so A beta peptides that is normally produced from the APP, and at physiological levels may play a role in like synaptic regulation and antimicrobial defense. And the problem arises when production exceeds this clearance, or when clearance mechanisms becomes inefficient. And we think when soluble A beta monomers, when their levels increase or accumulate, they tend to associate with oligomers or fibrils, and these are
Okay, but this is just the general mechanisms of how amyloid aggregation causes pathology. But okay, I would like to move to the next question. Thanks for your answer there. So in your study in this chapter, you only use male mice. So I wonder whether there is any reason for using only the male sex. And if you will be correlate this to the female, so what will happen in females? Do you think that will be similar or not? Or there is any evidence that perhaps there is no difference between males and females for the olfactory system?
The reason why we only used male mice in our animal studies is that female mice have these fluctuations in hormonal levels, so they're very difficult to do olfactory testing experiments. So that's why we only chose male mice. But in other studies, they show that there could be some differences between male mice and female mice, but the difference is not too big. So we think there may not be very big difference between this olfactory functionality between the sexes, but I think there will not be very substantial and significant difference between the sexes.
Okay, so thank you for that answer. Then now I'd like to move to the next question. So where you in your study have analyzed by immunohistochemistry.
Sorry, I have to interrupt you. We have to move on. We have
Okay.
15 minutes and we still have 3 opponents. So
Okay, that's fine. That's
thank you
okay.
very much for the interesting questions. And the opposition will be continued by Dr. Pishwa. He's an assistant professor in dimensional genomics at the Department of Psychiatry and Neuropsychology at Maastricht University. And he was also a member of the assessment committee.
Thank you very much, dear Prorector. I just quickly go, very nice presentation, very nice thesis, and very coherent. Compliment to you and promotion team. My question is regarding chapter 3, when you are in the biofluid and the nasal fluid biomarker, of course. And on page 74, you are showing some AUC curves, especially in figure A and B. then it is clear that the highest area under the curve belongs to the third quantile, while there is a drop in the accuracy or predictive performance when you go to quantile 4. So I would like you to comment on that what do you understand in the context of biological interpretations or methodological, what causes this?
Highly esteemed opponent, thank you for your kind words and your questions. So if I may restate your question, you're asking why quartile 4 group shows less power in predicting Alzheimer's disease. So we don't have like definitive answers to that, but our other results show that this third quartile group, moderate levels show the strongest association with like brain amyloid PET images or cognitive function, or in like this ROC curve analysis, this prediction is maybe that's because it's better reflecting since the nasal A-beta 42 levels are non-linear, so it goes up and maybe it reaches a plateau or declines, because with our interpretation, our inference is that because since we measured soluble A-beta 42, maybe since amyloid can form like oligomers or plaques or other forms of fibrils, so that the level of soluble drops down, but then it can on the same time insinuate that the amyloid pathology is worsening at the same time. So maybe that's why Q3, the moderate levels, is better reflection of the disease projection.
Excellent. Yes, thank you. But then my next question is that what did you make approaching in the fourth fixed quantile? Because another interpretation might be that the number of patients, like the cutoff that you make for the quantiles, is causing this, because maybe the high risk is the top 10% or something. And my question is that why did you choose not to use the linear or like the continuous measure of your biomarker assessment instead? That might give you a more accurate. Have you tried that?
Yes, my honest answer would be yes, we try to use this conventional correlation with the nasal A-beta 42 levels with like cognitive symptoms or brain PET amyloid PET results, but then the quartile analysis showed most strongest association with the brain PET images and cognitive functions. So that's why we chose this quartile analysis method. And
I assume that, no, that's understandable. But then that shows that your biomarker does not have a capacity of predicting at an individual level, and then you have to go for a population level based on the risk and that they have. But still, you could think of like having a top 10%, maybe that just like trying the different quantile stratification for that. Okay. So that's good. So I quickly go to my next question, which is related to my question is that you combine the neuropsychological as a panel as with your nasal marker, right? And then you reach a very high accuracy. What was the rationale behind that? What did you want to answer by doing that?
So your question is why we chose to combine this cognitive psychological battery test with our nasal A-beta 42 level analysis results. And the rationale was that we admit that the limitation of solely using nasal A-beta 42 levels as a means of diagnosing Alzheimer's disease. What we are trying to say is that with like electronic, maybe cognitive function tests in addition, and then with these routinely and non-invasive measures of basal A beta 42 levels combined with those two can strengthen this possibility of diagnosing A beta AD disease and
Right,
at-risk
yeah.
people. Yes.
Yeah, but shouldn't it be more logical that you start with the neuropsychological first without the nasal and then add the nasal to see that you will see any improvement, and then you can calculate that this improvement is independent information compared to the neuropsychological would have been maybe more interesting. Do you agree with me?
Yes, but we also try that analysis, but with the addition of neuropsychological battery test, it already has very superpower in this diagnostic power. So we chose this method instead.
And so I have a question regarding chapter 5,
Right.
but I'm going to
Yeah,
skip.
We have to
Yes,
move on
sure. Yes,
Yeah.
thank you very much.
because the opposition will be continued by Professor Kreun A. Jung. She is a professor of neurodegenerative disease and neuroinflammation at the Jung University in Korea, in Cheon.
Thank you,
Professor,
Director. Hi, thank you for your clear and very structured presentation. I have a few questions. In first one is AD patients research, and I wondered whether you assessed their sense of smell, the
So dear highly esteemed opponent, thank you for your question. So if I may restate, you're asking whether we measure the patient's olfactory testing. In this set of results, we did not perform this olfactory sensory ability in the patients, but in our another set of study, we performed this. Yes, we performed this olfactory testing, and we saw very significant drop of olfactory function in AD patients, but we did not publish this data yet. So yes.
Thank you. Second question is in amyloid PET research data. So, did you also look at the olfactory bulb or the olfactory cortex area in the amyloid PET? Because you analyzed the global several regions for the amyloid PET SUVR. So I just wondered, yes.
Unfortunately, our co-working hospital did not measure this olfactory bulb specific ROIs when measuring the amyloid PET scan. So we did not have the data for amyloid betas accumulation in.
Okay. In future, I think it would be quite interesting to see
Yes.
that. Okay.
Yes.
And next question is how about the tau in nasal fluid? Did you check it? In future plan?
That is also a very good question, and thank you for that. And yes, this result is also not included in my thesis, but in our ongoing study, we measured p-tau, but we did not quantify it because the level of p-tau was so in low concentration, it was not detectable using like quantifiable methods. So we used Western immunoblot method, and we witnessed p-tau is in nasal discharge fluid, but the results are yet to be published. Yes.
Okay, good. Next, in the ApoE genotype, the study, did you find any region-specific differences in the olfactory bulb or olfactory cortex between the ApoE2 or 3 and ApoE4 carriers? What kind of the differences depend on the ApoE type?
Okay, so thank you for your question. So in our results section, we saw that ApoE4 carriers, and they showed increased burden of amyloid and microglia in E4 status only compared to E2 or 3 carriers in the olfactory bulb only. So we think that ApoE is specifically more influential in olfactory bulb than olfactory cortex.
Okay. And last one is the human olfactory cortex is not a single structure. So which specific reasons were included or analyzed in your study?
Yes, that's thank you for your question. That's a very critical question. So we did not specifically informed about the specific region of olfactory cortex, whether it includes entorhinal cortex or amygdala, but based on in the Netherlands Brain Bank, there are very detailed experts. They guaranteed that it's from the olfactory cortex area, so we believe them, and that's why we chose, instead of specifying the olfactory subregions, we just named it as olfactory cortex.
Okay, thank you.
Okay. Then we move on to the last opponent. The opposition will be continued by Dr. Laurence de Nijs. She's an assistant professor at the Department of Psychiatry and Neuropsychiatry, and her expertise is neuroscience. Dr. de Nijs, there's some minutes left for you.
Yeah, thank you, Prorector. Dear candidate, congratulations for your presentation, thesis, and your promotion team. So I have a question now related to chapter 5, in which in the human olfactory bulb, so the postmortem tissue, you show that you have increased activation of microglia, and you hypothesize that this could lead to decreased neurogenesis, but you didn't show that. So I would like that you elaborate on how you come to that hypothesis, and maybe link that to the work you did on the 5XFAD mice there. Can you elaborate on that?
Okay, so highly esteemed opponent, thank you for your kind words and your question. So if I may restate your question, you wanted to ask if the glial activation we observe in the human postmortem samples and the potential consequences of adult neurogenesis, and as well as how this connects to the experimental evidence from our 5XFAD mouse model. And yes, we found a clear increases in microglia and associated activation in the human OB, and we think that although we do not test the cellular or molecular mechanisms of why these increases were observed, but we guess that this could be because of this OB's neurogenesis power and its very highly synaptic plasticity. Is maybe that could be the reason why there's high microglia burden in olfactory bulb. And linking that with the 5XFAD models, but unfortunately we did not measure this microglia burden in 5XFAD mouse, so we cannot compare the results with human postmortem and the mouse model models. But other literature's results indicate that 5XFAD mouse model may also have high microglia neuroinflammatory influences that can link to the amyloid pathology. So yes, but I do not have the definitive answers to your question, but maybe future more experiments can help to find answers to that.
And in your human tissue, how could you test whether neurogenesis is affected and the link to the microglia activation? Is there any way you could test that in that human postmortem tissue?
Yes, maybe we can use single cell transcriptomics to maybe, if microglia is alone doing some inflammation to related to amyloid pathology. Yes.
Okay.
Thank you.
Hey Jung, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return. So we are all there again. Jung, the degree committee here present online has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Jahan Shahi is authorized to confer upon this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Dear candidate, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I do.
By the authority vested in us, and by law and conformity with the decision of the committee here present online, I hereby confer upon you, Tahe Jung, the degree of doctor and grant you all the rights attached by the custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university shown by the below.
Thank you. Thank you.
Okay. Professor Jahan Shahi, you can now continue with the laudatio.
Yes, so we can be a little bit now relax. Dear Tahe, dear Dr. Jung, I hope I pronounce your name correctly. So again, I would like to congratulate you and your family and your loved ones on this wonderful achievement, earning your PhD. Your work represents the continuation of our collaboration with Professor Moon at Digest and follows the project started by your university colleague, Dr. Guvun Sun. I'm sure she's now watching the defense. She also defended at Maastricht University, similar to you, and she did similar work as you. After that project was completed, I visited Korea to discuss the next step with Professor Moon. During that visit, he told me about a highly motivated and talented student who was eager to come to the Netherlands and work on human brain specimens. That student was you. Soon after, we began preparing for your arrival in Maastricht. Already during the preparation phase, where we were drafting the brain bank application to obtain the tissue, it became evident that you are a good fit for the project given your clear insight on Alzheimer's disease pathology. Like every newcomer, you faced some challenges in the beginning, including finding a place to live, something many of us here can relate to. But you handled it out very well, quickly settled in, and immediately started your project. Unlike most PhD trajectories, which usually takes about 4 years, and the students have sufficient time to adapt to the new environment, you had less than a year to complete your project. That is very demanding task and require not only scientific knowledge, but also focus, discipline, and resilience. You demonstrated all those qualifications from very start. You quickly established yourself as an independent and reliable researcher who could manage her work with confidence and independence, something every supervisor dreams of. Your maturity, dedication, independence, and of course your intelligence have all been key ingredients to your success. The result of your work are excellent, as reflected in your thesis and in the manuscript currently being revised for publication in a respected journal. These achievements clearly show your scientific capability and commitment. I'm sure your ambition will not stop here. I hope this PhD serves as a strong platform for you to continue developing your career wherever it may take you. With your skills, your curiosity, and your work ethics, I have no doubt that you will succeed in whichever path you choose. On a more personal note, I have always been fascinated by Korea as a nation and Korean culture. This was one of the reasons I always eager to collaborate with. I've been always eager to collaborate with Korean institutions and Korean researchers. Since then, I have never regretted that decision. And after seeing your excellent work, I'm even more motivated to continue this collaboration in the future. So once again, congratulations, Dr. Jung. You can be very proud of yourself. And I wish you all the best in your future career. And I truly hope that our paths will cross again.
Thank you.
Thank you.
Okay. So I give the board back to Prorector after Lodos.
Yeah. Thank you very much, dear Dr. Jung. Also on behalf of Maastricht University, I congratulate you with the degree you have acquired, and I hope you will continue your successful research in Alzheimer's disease. And hereby, I declare this ceremony ended. Thank you all, and please keep online for a few seconds. Then you can congratulate Dr. Jung.